Okay, so here we're gonna introduce a very important yet useful program for RNA-seq data analysis, and that is the Cufflinks packages. So after using TopHat to align the reads to the reference genome, you might then want to further infer the abundance of the transcripts. This is exactly what Cufflinks does. It accepts aligned RNA-seq reads and assembles the alignments into a set of transcripts, and then it estimates the relative abundances of these transcripts based on how many reads support each one has. There are 4 packages in Cufflinks package. They are Cufflinks, CuffCompare, CuffMerge, and CuffDiff. Cufflinks and CuffDiff are 2 major parts of this package. Cufflinks assembles the fragments and calculates the transcript abundance, while CuffDiff calculates genes and transcript expression level in more than one conditions, and then tests them for significant differences. In this video, we will focus on Cufflink and CuffDiff. There are 2 major functions in Cufflinks. They're fragment assembly and abundance estimation. It will assemble the aligned fragments from TopHat and then estimate the abundance level of different transcripts. Now let's take a look at how Cufflinks works. With pair-ended short fragments as input, we want to know the distinct splice mRNA isoforms. By identifying the incompatible fragments, we could get an idea on what the different isoforms are. Like the yellow, blue, and red fragments here, they must come from different mRNA isoforms. Then with these mutually exclusive fragments as the starting point, we connect all the other fragments by overlapping. In this example, the yellow, blue, and red represent 3 possible transcripts, and we try to assign the fragments to either one of these 3 transcripts. The key idea here is the number of mutually incompatible reads is the same as the minimum number of transcripts needed to explain all the fragments. After determining the transcript isoforms, we need to assign each fragment to different transcripts. For example, we want to determine which isoform the violet fragment comes from. It may come from the blue or red transcripts, but it's less likely to have such a long fragment in the case it comes from the red transcript. So, based on the knowledge of the fragment length distribution and also the likelihoods function, we could get the most possible model for us to get the reads, actually. And after assigning each fragment to each transcript isoforms, we can proceed to calculate the relative abundance of each transcript by FPKM. The full name of FPKM is fragments per kilobase of transcripts per million fragments mapped. It takes into the fact that the number of reads is proportional to the lengths of transcript, as well as the total number of reads. Therefore, after such calculation, we could compare the transcript abundance across different transcripts and also different experiments. So after introducing the basic ideas, let's take a look at how to actually run the codes. It's pretty simple. All we have to do is to assign the output directory as well as the input file from TopHat. And that's it. So now let's proceed to CuffDiff.
After running Cufflinks itself, there are a few additional analysis packages that can be used. CuffCompare compares the Cufflinks output, the transcriptome assemblies, to a reference gene list. It is able to identify novel isoforms and novel genes. To run CuffCompare, first make a text file with the names of all transcript.gtf output files from Cufflinks. This is the first input, and the second input is a reference GTF file for your transcriptome of interest. CuffCompare outputs many files. there is one combined.gtf file with the union of the transfrags across the samples. There is also a tracking file, which matches the transfrags between the cufflinks outputs, which is helpful if you do not do the assembly with a reference transcriptome, as the same transcripts may have different identifiers between the samples. Additionally, for each input file from the individual cufflinks runs, you get a refmap file showing for each reference gene which transfrags in the sample match that gene. There's also a tmap file showing for each transfrag which reference gene is the best match to it. Another program is cuffmerge, which creates a merged GTF file for many samples. It also automatically runs cuffcompare. The combined GTF file can then be used as the input for cuffdiff. To run cuffmerge, again make a text file with all cufflinks output GTF files. The arguments are a reference GTF file, a FASTA file for the genome, which it uses to exclude repeats and classify the transfrags. You also give it an output file prefix and the assemblies.txt file. The output is the combined GTF file. Cuffdiff is a program that quantifies expression levels in multiple samples and tests for differences in expression between the samples. The input is the combined GTF gene file from cuffmerge, as well as the SAM files from each individual cufflinks run on each sample. It is able to test for expression differences between genes as well as between isoforms of genes. To run cuffdiff, the arguments are the output prefix, a FASTA file of the reference genome, labels like the sample name for each sample, the combined transcript GTF file, and then for each sample, comma-separated list of the accepted hits.bam file for each replicate.
Cuffdiff outputs many different types of files. There are two main groups of files, tracking files and differential expression test files. There are also a few other tests like differential splicing, CDS usage, and promoter usage. This makes it hard to keep track of which ones are which. Hopefully this section will help in figuring out what files are more useful for your analyses. There are three types of tracking files outputted by Cuffdiff: FPKM, count, and read group. Within these three types, four subsets of the transcriptome are quantified. These are isoforms, genes, coding sequences, and primary transcripts. FPKM tracking files basically quantify the expression of these subsets. Count tracking files estimate the number of fragments that originated from each subset, while read group files contain the expression and fragment count for each subset in each replicate. The isoform tracking file tells us the expression values of unique transcripts. Genes groups the unique transcripts by gene and shows us gene expression. The coding sequence or CDS file groups transcripts that contain the same coding sequence, which can serve as a proxy for proteins. The TSS group groups transcripts sharing the same transcription start site. Here is a nice schematic by Stephen Turner of how these transcripts are grouped in these files. Isoform files do not group transcripts, so A, B, and C are distinct. In the gene subset, A, B, and C are all grouped together because they are from the same gene. In the coding sequence subset, B and C are grouped together because they share the same protein output of exon 1, 2, and 4. And in the primary transcript subset, A and C are grouped together because they have the same inferred transcription start site. Differential expression tests show whether expression is upregulated or downregulated significantly between samples. Like the tracking files, they output for isoforms, genes, primary transcripts, and coding sequences. Differential CDS analysis is only performed when all isoforms of a gene have a coding sequence attribute. Useful fields in these files include test status, p-value, q-value, and significance. The test status can tell you if the test was successful, if a test was not done, or if the test succeeded but the result may be compromised. Q-value gives you the multiple hypothesis FDR-adjusted p-value, and significance gives you a simple yes or no answer to whether this differential expression is something noteworthy. Cuffdiff also outputs differential splicing, coding, and promoter use tests. Differential splicing gives you information on how much differential splicing exists between isoforms processed from a single primary transcript. Differential coding output tells you how much differential CDS exists between samples. This one is particularly useful if you're examining proteins since it measures output post-mRNA processing. Differential promoter use looks at the amount of overloading detected among its primary transcripts and compares that between samples. Usage of alternative gene promoters drive widespread cell type, tissue type, or developmental gene regulation, and aberrant use of alternative promoters have been found in disease-associated genes.